So, welcome to video 6 on G08, where we're going to talk about metagenome dereplication. So this is a particular problem where we have a lot of contigs and a lot of mags that are generated from our metagenome assemblies. And in this case, it's an issue where we have co-assembled it together and we're trying to bin them appropriately. And if we have a small, in bacteria, small plasmids or small contigs, they can be difficult to categorize. And we have a lot of closely related mags, but that do have some sequence diversity. And what dereplication seeks to do is to reduce the number in an effective manner, so that we have representative contigs, but not many very similar contigs that might give us a biased result. So one way of doing this is use the average nucleotide identity, the ANI, and more effective approaches are needed than our typical approach of pairwise alignment. So these genomes are clustered together using MASH, which you see at the start here. These are metagenomic assembled genomes, and we are able to cluster them and use different approaches such as BLASTn or MUMMER or others to allocate them into groups, so we can identify what is the most representative data set. So typically per group, there'll be just one mag is used to represent them. So it's again dependent on the thresholds that you are interested in. The choice of tool will affect the result as well. And this will also affect the abundance estimates because we have different mags that are present. And we want to remap our reads back to these representative mags only to estimate their relative abundances. So therefore, we need to consider what threshold is being used, not to bias that estimate too much. And what is blatantly obvious is that this is quite tricky, and our true abundance estimation is basically a function of the ANI threshold used. So by using multiple thresholds, that can help give you more consistency over what's actually an accurate estimate of the true abundances. So what will dereplication will do, we're trying to identify the highest quality representative genome or contig per group. Derep is good at this. It's fast and efficient. It uses min-hash distance in MASH to estimate the similarity, but depends on the metagenomic completeness. And it uses other metrics related to the ANI, GANI, and AIM. And it requires the metagenomes to be at least 50% complete. An example of how this works is shown here for Staphylococcus aureus, where you have the whole genome assembly around in a circle in black, and then we've got a co-assembled one in orange, which you can see is a reasonable representation, but there will be some conserved regions that are shared with other organisms. Inside C, we have a portion of our wider diagram B, where we see the gray contig here, that stretch, and a portion of the orange co-assembled one is we have a zoom in shot where we can see that we can remap the reads back to a gap in the alignment where the co-assembly did not work. So co-assembly is pretty good, but we can also find diversity within there. So that concludes this part on dereplication. Please see the next video on assessing metagenome assembled genome quality.